Peter Marks is the Director of Center for Biologics Evaluation and Research at the FDA, better known as CBER. CBER is responsible for the regulation and approvals of all gene therapy and cell therapies, including the accelerated approval of Sarepta's Elevidys. And Peter has been instrumental in listening to the patients, listening to communities, and understanding how much we need to get these therapies into our kids. So I'm very grateful for his work and extremely honored to have Peter Marks, Ph.D. here today. Thank you.
Thank you. Thanks so much. Thanks so much, Deborah. Thank you so much to the Millers for hosting this, and thank you for coming today. So what I will try to do over the next half hour or so is tell you a bit about what we're doing at FDA to try to advance gene therapy, try to tailor it a little bit towards DMD. But we at FDA are trying to help advance the progress of gene therapy. We're trying to help deal with existing issues with product development and trying to address challenges in gene therapy development. And I'll tell you a bit about that as we go forward here. So there are real importance to us for all diseases that are rare. It turns out that as we've gone through, and now that we know the entire sequence of the human genome, and we have a better understanding of medicine, we now know that there are at least 10,000 disorders that are rare, that are hereditary or acquired. And many of these can be addressed using novel therapies, potentially, and potentially gene therapies. And addressing these is very important because, A, in aggregate, there are millions of people in this country affected by rare diseases. You are some of them. And additionally, until we can understand how to treat these rare diseases, we won't really understand how to use gene therapy on larger populations. So this is important for us, not just for rare populations, but for everyone. Now, in rare disease, there are real challenges sometimes in developing drugs. We have this issue that in some cases, natural history information may be limited when there are very small populations. In some cases, there's a diversity of disease manifestations. We know that even in DMD, right? That some people progress at different rates. Sometimes that's related to the gene mutation itself. Sometimes it's just related to the fact that people are different. It's hard sometimes to study rare diseases because the time course of change can be very prolonged. And although to an individual, it may not seem to matter, but when companies are studying things, we generally study things over the course of 6 months or a year. It's hard to study things over many years, and so that's a challenge. And in some cases, for diseases, when things are lost, they're not regained, and it's not. We don't have ways with gene therapies to restore them, so showing that something doesn't change, that something is stable, can be challenging, and that's often what we're trying to do. Now, to treat rare diseases, there are a variety of things that we can use. We have small molecules. Those are the drugs that we take, like when you take an aspirin or acetaminophen, Tylenol. There are protein therapeutics. Those are things like growth factors and that are given. There are antisense oligonucleotides. We're familiar with those in the DMD space. Those are things like exon skippers. And then there's gene therapy. And I'm going to talk to you today about gene therapy. That's not to say that the other therapeutics here aren't really important. They are. It's just that gene therapy is quite an exciting area. For gene therapy, and I think just so you know, the size difference here, I think, was lost in translation, but they shouldn't be, disadvantages shouldn't be so small. Advantages, there's kind of an inadvertent, I don't know how that happened. It shows you what happens with artificial intelligence. Advantages here the idea here is that the ability to have a therapy where one dose of something could provide long-term benefit or possibly even cure, that's huge. And you know, even in the absence of everything else, not having to take medicines on a daily basis is a big plus, but when it could potentially make a huge difference in the progression of a disease, it takes it to a whole nother level. Another advantage of gene therapy is that unlike some other medicines, small molecule drugs are kind of challenging. You discover them and you try to see if they work, and any given one only has a very, very small chance of working when you start to study it. Whereas gene therapies, genetic therapies, have a much higher probability of success. And because, as I'll show you, one can use somewhat of a platform approach, you can address a lot of different diseases using this approach. So that's kind of an exciting part on the advantage side. The disadvantage is that right now, as people in this room have probably read in the newspaper, these are expensive therapies. They're very expensive to make because gene therapies are made using cells that help make the actual viruses that deliver them often, or they involve a lot of steps in manipulating cells, and that gets to be very expensive. Plus, there's the cost of development, and plus other commercial costs. And then there's the potential, obviously, that they could have irreversible side effects. That's our job to try to make sure that we minimize that possibility. That is toxicity on the genome, and we have to make sure that we're vigilant for that. There are also things that, at least at this point in time, have to be administered at specialized centers. If somebody at a local health stop says they're going to offer you a gene therapy, just say no, because they need to be administered in specialized centers, and that's why we have experts who administer them to take care of some of the downstream complications that can occur after they're given. And in that way, they can be administered safely. And one of the other things that is an aside is it is a new business model for companies and for insurers, so we're having to negotiate this new environment. Now, the two major ways that gene therapies are delivered are we can take cells from the body and manipulate them in a petri dish, essentially, outside of the body, or we can administer the gene therapies directly to somebody. In the realm of cancer therapies, a lot of them have been produced using this outside of the body approach, the ex vivo approach, in which cells are taken from an individual, modified in a petri dish, grown up, and then given back. And those have used a set of vectors called lentiviral vectors related to retroviruses. And then on the other hand, the directly administered gene therapies, which are given either locally or into the vein, that includes the existing therapy for DMD, those are often using a viral vector called adeno-associated virus. Adeno-associated virus is it's something that circulates. It doesn't actually cause a lot of problems in people, and that's why it's such a good one to use for gene therapy. But it has its limitations because it can only carry genes of certain size. It basically, if you try to overstuff it, it gets unhappy. But it has become a very good gene therapy vector. We're also now seeing new methods of delivering gene therapy, and I'll save those for the end, to kind of looking forward. The other piece that you may have heard about, and I think it's probably worth starting to educate people about this because we're going to see these come to pass, possibly even into the DMD space in the not too distant future, is genome editing. Genome editing is something that's been around for decades but became a reality in the past decade as we had a new discovery of a molecule that's able to clip DNA and allow it to potentially have a new piece of DNA inserted or to inactivate a gene at the spot, and now it's gone even further. And so over the course of time, this kind of approach using, sometimes people like to consider them molecular scissors, that's why there's little scissors there, has become a very potentially powerful approach because if you can repair the mistake in the DNA itself, you have a much larger number of mutations that can potentially be addressed, and potentially one could lead to curative effects and get over some of the challenges that we currently have with our current version of gene therapy. The course of time now is really quite impressive because since the discovery of CRISPR-Cas9, this genome editing system, which was discovered by someone who won the Nobel Prize for it ultimately with a colleague, Jennifer Doudna, in the course of about a decade, it went from being discovered to being used to make a product that's now approved. And we now have 19 approved gene therapies in the United States. There's a color coding here. The blue ones are those cell-based chimeric antigen receptor T cells that are mainly for cancer. The green ones are genetically modified stem cell therapies, including one that was made using CRISPR-Cas9, which is for sickle cell disease and beta thalassemia. And the purple ones are directly administered gene therapies, and you'll note the DMD therapy, Elevidys is there. Just so you know, these things are given, the Elevidys is the brand name. I'm obligated to show you the international non-proprietary name, which I'm not going to pronounce for you because I don't want to embarrass myself. But I'll try. Delandistrogene moxaparovec, and the four letters at the end are a technicality. But this is the gene therapy that we approved last year for DMD. It was given an accelerated approval. Accelerated approval is a pathway that the FDA can use to approve something earlier than it would based on a clinical endpoint of how someone feels, functions, or survives based on a biomarker or an intermediate clinical endpoint. And a biomarker is basically a laboratory test, and an intermediate clinical endpoint is one that it could be something just short of how someone feels, functions, or survives. And the reason why I have these little graphics here is to show that it was a small study that we used. This was, the initial study was based on about 40, the initial submission is on about 40 patients. And in that study, the overall population did not have the kind of statistical significance that we needed to see. But age 4 and 5, on the right-hand side of the screen, you can see those error bars don't overlap. When you don't have overlapping error bars, that usually means that there's statistical significance, and that allowed us to give this an accelerated approval. In the coming months, you'll hear more about this as there are submissions pending. But the bottom right-hand box just shows that there was also a correlation here in the 4 and 5-year-olds between the amount of microdystrophin, the gene therapy product that was produced, and the outcome. So that's something also that makes us feel like the totality of what we saw allowed us to give this an accelerated approval. And eventually, once we see the clinical endpoints that we need, it can potentially convert over to a full approval. Now, gene therapy is currently at a critical juncture due to a combination of factors. And although it's extremely exciting to see all of the scientific development, we've gone out ahead of ourselves with our science, and our manufacturing technologies have to catch up. And so one of our jobs at FDA is to help manufacturers develop the methods and the tools to be able to produce these gene therapies more efficiently. There are also issues in trying to make sure that enough products come through the development pipeline, because when things take a long time to get through development, it's just a fact that investors don't have a lot of patience. If I could wish one thing, it would be that venture capitalists get like really patient with development, but they don't have a lot of patience, and so they want to see return on investment quickly, which means that if we can help speed up clinical development timelines, we'll help more patients because more people will invest in making these products and bringing them forward. That's one of those places where the needs of patients and the needs of investors actually are somehow aligned. Go figure. We also have an issue, which is that if one looks across the globe, there are different global regulatory requirements which sometimes prevent people in other countries, or sometimes in this country, from getting access to important advances, and we're trying to address those issues. So I'm going to tell you about, for the next 10 or 15 minutes, a little bit about what we're doing to advance the manufacturing of gene therapy, what we're doing to work towards more clearly defining the use of accelerated approval for gene therapy, because accelerated approval, this ability to not have to wait for a final clinical endpoint to give something an approval, is very important in this area. We'll talk about what we're doing to try to address the issue of working with other regulatory authorities, and also talk about some of the things we're doing to try to help further speed up the development of products. So in the area of manufacturing, it's probably, you might say, why should we care? Well, the reason why we care is because a lot of the innovation that comes in the area of gene therapy comes from academic laboratories. That innovation is so important. The problem is it has to be translated into commercial processes in order to be of benefit to people. And making that transfer is sometimes very expensive and time-consuming. And that's because academics are great. They like to do things. They do them relatively rapidly in their institutions using local protocols. The problem is that commercial processes have to be done in a standardized manner, and making that transfer can sometimes cost millions of dollars and take a year or two. So we're trying to work in a collaborative project with the Bespoke Gene Therapy Consortium, which is sponsored by the Foundation for National Institutes of Health, to find kind of a playbook so that we can help that transfer occur more seamlessly. The other thing we want to do is see if we can make more of these processes automated, because it turns out that having people in large facilities making these gene therapies ends up costing a lot. And if we can automate some of these processes, it will reduce cost and probably also speed us towards a place of having more consistency in manufacture, which would be a good thing. So at some point, we'll hopefully see devices that are able to do at least part of the manufacturing for gene therapies. But something that those are things, manufacturing is something that we can, we don't do it per se. We're not manufacturers at FDA. We do some research in this area, and it's an area where we can work with the ecosystem to try to move forward. The area we do have some real control over is what we do about our regulatory approvals, and we can leverage accelerated approval to help bring things forward. Now, one of the really nice things about gene therapy is that when you understand what gene you're actually working with, you already have kind of a one-up on a lot of other types of development, because you have a tool which you at least know what to look for. If you're replacing a gene that's missing, you know that you need to look for the expression of that replaced gene. And that means that we can look at things like enzyme activity, structural protein levels, and correlate them with clinical endpoints in either animal models or human systems, and get an idea if something is reasonably likely to predict. That's our regulatory standard for accelerated approval. If a biomarker or an intermediate clinical endpoint is reasonably likely to predict how someone is going to do after they get the therapy. And we're lucky, in some cases, we have animal models that mimic human disease. It's not always the case, and not every animal model is perfect. But when one does have an animal model, and the animal model is, for example, deficient in a given protein and has a deficit, and then you can give a gene therapy and show that the animal is better, that is one way of understanding if you can get to a similar level of expression of that protein in a human, you can say perhaps that it's reasonably likely to predict. Animal models in DMD include, for instance, a dog model. In some case, we have rodent models. And then there is the human observation. And that actually also has been relevant in DMD with Becker's, which is that some people don't have complete absence of a protein. They may have an abnormal protein or less of a protein. And if you can show, again, that you can restore the level of a protein through a gene therapy to a similar level where somebody is able to have no deficit or minimal deficit or less of a deficit, one has a way to reasonably likely to predict. Either way, these are ways that we can get things to a place where it's a fully approved product based on a biomarker or an intermediate clinical endpoint, and people can start benefiting from them. And then over the course of time, it could be years, we get the clinical evidence that allows us to give something a full or traditional approval. Sometimes we might not get that evidence, in which case we might have to take something off the market. Sometimes the evidence may come in and we might say we need to do another trial. But this way of using accelerated approval at least lets us get products to people more rapidly. And in the rare disease space, it's very, very important. You'll probably hear a lot of noise about accelerated approval in the coming few years, because there are some that don't like this pathway because for larger indications, they view it as a way of kind of getting around the FDA's need for substantial evidence of effectiveness, which is our regulatory standard. And I would say maybe that's true for like a drug that treats high cholesterol, but where maybe we should be pretty rigorous. But in this area of rare diseases, people need stuff. I mean, I think everyone in the room would agree, we need something now, not later. And so if we can bring things up and move them up with this pathway, it's important to use. So a couple of other things we're trying to do. You know, although we are responsible at FDA to the United States public in terms of what we do, we're also interested in trying to the extent we can, also to help globally with public health, and to also reap the benefits from work that's being done in other countries. And toward that end, we are trying to reduce global regulatory differences because we believe that ultimately it will benefit people here in the United States. And let me take you through that thinking, because sometimes this is what happens when you spend too many hours awake. You sometimes get deranged thinking like I do, but let me take you through that. The idea is that if we have an ability to have better global regulatory convergence, certain products that don't have a large enough need in the United States because there aren't enough patients will have enough need if they're needed in multiple countries. And so if we had a regulatory environment that allowed sponsors to use one application and take them to multiple countries, we would potentially get them on the market here in the United States. They'd get a market across the globe, and we'd actually have all of our different populations benefiting from them. The other thing that's potentially helpful is if we had a system whereby we had better regulatory convergence, each regulatory agency wouldn't have to review these products, and it wouldn't quite take as much in the way of resources. But we really want to see, again, ways to make sure we see more products getting to patients in need. Another thing that we're trying to do is help small companies and some large companies get products to the market more rapidly. Does anyone here remember something called the COVID-19 pandemic? Right, okay. So, you know, as part of that, the areas that we regulate, although it includes gene therapy, also includes vaccines. And during the COVID-19 pandemic, I spent a lot of time with vaccine development. and dealing with Operation Warp Speed. Operation Warp Speed was a way of trying to speed up vaccine development, and it probably shaved about a year off of the time that it took us to get to COVID-19 vaccines. A fair amount of that time savings came from the fact that we had very good communication with companies in a way that we normally don't do at FDA. Normally, the way FDA has communication with companies is the company says, oh, we want to schedule a meeting. We say, okay, what kind of meeting do you want to schedule? And depending on what kind of meeting it is, it takes 30, 45, or 75 days to get a meeting. And that means that, the cycle time is pretty long. What we did in Operation Warp Speed is said, no, we're not going to do that. When you have questions, you're just going to have a project manager, and you're going to call them, and we're going to set up a meeting right away. And we believe that in this rare disease space, if we move from having meetings that take 2 to 3 months to schedule to 2 to 3 days to schedule, we could again get more products to people more rapidly. And probably in the next couple of weeks, you'll hear the announcement of the first set of products that we're putting in this pilot in the pediatric rare disease space at our center to see if we can show that this makes a difference. If it makes a difference for a couple products, we're going to try to expand it to all of the products in this space. It's just that we can't do it immediately without showing that it works because it takes a lot of extra effort to do. So we have to show our Congress and industry that it actually makes a difference. Finally, I'd like to talk about something that really excites me about what we do at the Center for Biologics. We have kind of an interesting history at the Center for Biologics. Biologics in the United States were regulated through the National Institutes of Health until 1972. And so we have a very strong scientific bent. And there is an area of regulation where regulation and science come together very nicely. And I'll just tell you about this because as I tell you about this, it will help us understand where this field of genome editing is going and some of how we're thinking about how we'll be regulating it. So gene therapies are a form, potentially, of platform technology. Unlike small molecule drugs or protein therapeutics, where each one isn't all that related to the other one in most cases, gene therapies of a given type, adeno-associated viral vectors of a given serotype, are closer to one another than many other medical products, and they only differ by the different gene that they deliver to the person. And that's what's meant by these different colored boxes, small boxes in this. So this concept, though, means that the fact that you're reusing something over and over again, much like a razor handle and a razor blade, the new piece of gene segment is like the razor blade. The razor handle, which is the same over and over again as the gene therapy vector, means that we don't have to reinvent all the information for the razor handle each time. We can use that over and over in some cases and only focus on the differences of the razor blade or the gene that's being put in. That may sound like, why does that matter? Well, it matters a lot because it speeds up development by not having to have us go through obtaining all of the toxicology, manufacturing, control information, everything else that has to go in when one goes from one gene therapy to another. And that might also speed up development times and reduce cost. And so we're lucky enough that last year Congress passed a part of an Appropriations Act that allows us to designate things as platform technologies, which allows manufacturers that have an approved product to leverage them as a platform technology so that they can then rely on information from one product as they move to another. Now I'm going to try to show you how this can be applied to something that's very cutting edge. So I mentioned CRISPR-Cas9 genome editing. This is quite a remarkable thing. I mean, it's basically a macromolecular. It's a big set of molecules, essentially, proteins, attached to a segment of genetic material, a guide RNA, that is able to identify a place on DNA where it sits down and it clips the DNA. And then at that clippage site, in the first iteration of this type of gene editing, either it can inactivate a gene, so that if there's an abnormal gene that's going on that you want to stop, it can do that, or it can open up the DNA so you can put something in. Now, this is the first generation of these products, and it really is something that now has been built even further, because one can express this in an adeno-associated virus if one wants to. Alternatively, one can make these constructs as part of mRNA, something that I won't go into all the details, but we got a lot of familiarity with during the pandemic. You might have heard of the mRNA vaccines, and can make essentially little packages of the gene therapy in lipid nanoparticles, which are essentially little fat globules that contain the construct. And by doing this, one can then essentially direct this gene therapy to a location. Right now, most of the time when you don't direct it, it goes to the liver, but people are working on putting surface studding to these little fat droplets that will take them to other places in the body, like to the muscle or to the brain. And so this is quite exciting, and the technology is advancing relatively rapidly, such that I suspect we'll see products made like this on the market within the next 5 to 10 years. What's more is that the original version of this CRISPR technology wasn't perfect, because it clipped both strands of DNA, which left 2 hanging ends. That's never a good thing, because it could lead to untoward events and inadvertent mix-ups. So people have worked diligently and have advanced this technology very rapidly, so that there are now versions of CRISPR that don't cut both strands of DNA. They just cut one strand. That means things can get less mixed up. And then they've taken it one step further. They've made it so that they've joined this system to something that works almost like, it's the best equivalent I can say is it's almost like having a word processor. By putting a reverse transcriptase onto this construct, you can actually insert sequences of DNA, at least short sequences right now, 100 or 200 base pairs into the genome. And there are now people that have worked on this, so you can actually put a site in and put in large segments of DNA, such as possibly even a whole dystrophin gene. So it's quite exciting here where this has gone. The reason why this is all so important is because as we look to the future, I think we may see ways of producing these products much more readily than the current generation of adeno-associated viral vectors. It may have the advantage also that, unlike adeno-associated virus, which sometimes is a problem because if one has been exposed to the virus previously, one may not be able to be dosed with the gene therapy, at least at this point in time, although people are working on that. This technology doesn't have that same problem, and it also potentially has the advantage that it could be redosed. So really an exciting thing coming up. Not a reality yet for these prime editors in terms of products, although there is, as I said, one CRISPR-based cell-based gene therapy already on the market. So this is something that could become a very real technology, bringing benefit to patients in the coming years. So what I've tried to do is tell you about some of the work that's been going on. Our goal at the Center for Biologics is to try to bring this power of genetic medicine to as many people as we can, as quickly as we can. I think the important difference that we've tried to come to over the past years is to realize that although we're a regulatory agency, the regulations have to ultimately serve getting products to patients. And so we're trying to focus on the patient. and use what we can to negotiate the regulations to get there as rapidly as possible. So I will stop there, and I think there might even be time for questions. Thanks. If yes or no. Maybe we just have extra time.
Thank you so much, Dr. Marks. Are you willing to take a couple questions?
I'm willing to take a couple of questions.
Okay. Do we have a couple questions here?
There we go. Right, that's a great question. So the question is, what's the difference between an exon skipper and a gene therapy? The exon skippers are generally, they are a different class of molecules than the gene therapy, so it's a great question. Exon skippers are medications that generally need to be given repeatedly because they're not changing the genetic material, they're just allowing the material, the machinery of the cell to make a certain change, not permanently, but essentially to produce a protein on an ongoing basis by giving them repeatedly. So exon skippers are drugs that are given on a repeated basis, whereas the gene therapies are given once, hopefully, or very few times, and they actually change the genetic material itself by either altering a gene or adding something back in to do something once and for all. So right now, there's probably a combination of these. There's nothing that says that one can't have someone get a gene therapy and then potentially get an exon skipper. All sorts of combinations are possible, but they are different things. That's the best.
Excellent. You have a very good way of taking very complex science and making it understandable. Thank you. Are there any other questions? If not, then I want, again, a round of applause for Dr.
Thanks.
Marks and for the work that he's doing for all the rare diseases.
I'll give you the clicker back. Thank you so much.
Oh, we had a question. We don't see the question.
There we go.
Okay, shout it out.
Hello?
We got a microscope.
Hi, I have a quick question, actually a big one. When we talk about exon skipping and the mutation that is there are numerous mutations when it comes down to Duchenne, and it's very complex. And when exon skipping products are being developed, there's a cost attached to it. My question is, can we look at some kind of a protocol or a platform where multiple exons, even if there's a single exon skipping which is being developed, can be applied to other exons without the cost being as high as it's? Because if you run into preclinical studies or even dog and mouse, it just becomes too expensive. And then there are companies who would not be probably interested in taking that up for rare exons.
Yeah, I'm happy to just say you can keep that, I guess. From a technical perspective, I totally see where you're coming from, and it potentially could be considered platforms. Just so you know that our center doesn't regulate exon skippers. They're regulated by the Center for Drugs, so I'll take that back to Peter Stein, who was here yesterday. But it's a good question. It's a very valid question. Now I'll say one last call, going once. Oops, looks like one more. Maybe we'll take that as the last one.
Thank you for the detailed explanation. That was very helpful. The CRISPR stuff, how is it administered? Also through AAV? I've heard that some studies are also using AAV. So if my kid has an antibody to an AAV, how is CRISPR even a possibility?
Yeah, so it's a great question. So CRISPR is being, some CRISPR, some people developing it are using AAV, but some are developing it using the lipid nanoparticles. So the idea here is it doesn't require a virus. And so the gene therapy is administered essentially in what's the equivalent of that fat bubble, so that people don't have immunity to it. And so potentially, this is actually, to me, exciting, because potentially if in the event that some of our AAV-based gene therapies, people who have gotten those now, if for some reason they wear off at some point, there's no reason potentially why CRISPR, given in a lipid nanoparticle, couldn't be used subsequently, down the line once those products are developed to salvage that. So I think that's an exciting place to see work going on. Thanks. Great.
Thank you.
Thank you so much.
Thank you so much. And actually, your questions were a really good segue into the next session, which is on gene therapy. And when Håkon was diagnosed, there were virtually no programs that were in review at the FDA. And we're so excited to see just a plethora of products going through approval process with the FDA. We like to keep them busy. And the way that Cure Duchenne does that is we take contributions from different donors, small donations. We aggregate them together, and we invest in these companies at a very early stage and a very crucial stage where oftentimes they just need to prove a point. They need to get a proof of concept to see that there's a there for Duchenne with their technology, with their science. And so many of you have supported this research that is actually turned into drug development programs and are actually approved drugs now. The way that we are able to make smart decisions is, I'm a little biased, but we have the smartest and best scientists in Duchenne working for Cure Duchenne. Dr. Mike Kelly, our Chief Scientific Officer, has been with us for 13 years, guiding our investment and research strategy. Liana Orlando has been with us for about 5 years, and Poting Liu's been with us for a little bit over a year. Together as a team, they have led the Duchenne field in terms of investing in really meaningful science. And right now, it is my pleasure to introduce Dr. Mike Kelly, who will introduce the gene therapy panel. Thank you.